Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme Acetylcholine Esterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis.
Okay, so I got a little antsy there on stopping that video. The last couple of words of that was acetylcholine esterase is breaking down the acetylcholine into its 2 components, and the choline component is being taken back up into the neuron so that it can be used again. So let's do a quick recap of some of the things that we talked about on that video. So let's look back to the previous slide, I hope, here anyway. There we go. Okay, so remember, this is a nerve cell. It's called a motor neuron because it's going to stimulate a skeletal muscle to contract. Another thing I want to quickly recap is that we talked about cell membranes being polarized, and when you depolarize a membrane, if you depolarize enough, then that cell can do its job. So the job of a neuron is to stimulate another cell. The job of a muscle cell is to contract. So when each of these cells are depolarized enough, that's called their threshold, or you reach the minimum threshold of depolarization, then that cell is going to work, do its job. So with that said, we've got to have depolarization of this axon. Now remember, this axon is going to come down here to an axon terminal, and then there's going to be a tiny little space between that axon terminal and that motor neuron. That space is called the synapse or synaptic cleft, and that part of the muscle cell membrane is going to be called the sarcolemma, or that part of the sarcolemma is the muscle end plate. So now remember, we've got depolarization going on along that axon until it reaches the axon terminal. Now we're really not going to talk much more about that nerve cell right now because we're going to cover nervous system in a later chapter. But it is important in that we have to understand that nerve impulse is traveling down in a wave of depolarization. So this membrane is depolarizing, 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 depolarizing. And when that depolarization reaches the axon terminal, voltage gated channels are going to open here that will allow calcium to come from the extracellular environment into the intracellular environment. That calcium then stimulates the cells to take acetylcholine, package it up into vesicles, and then through endocytosis release it into the synaptic cleft. It then diffuses across the synaptic cleft. Okay, now remember, this is our muscle cell, the sarcolemma, that has acetylcholine receptors. So let's look at the next slide. Here we have the synaptic cleft. Here's the sarcolemma with the acetylcholine receptors. So acetylcholine then binds to the receptors on the muscle cell membrane. Acetylcholine is a ligand, which means it works sort of like a key because these receptors, the acetylcholine receptors, are ligand-gated receptors. So you can simply think of it as the acetylcholine is the key. When it's in the receptor, the gate will open. When that gate opens, acetylcholine does not go in. These are sodium gates. So sodium, remember, is in higher concentration outside the cell as compared to in. So the sodium rushes into that muscle cell. Also remember, sodium is positively charged. So if the inside of this cell was more electronegative, as that sodium is coming in, positive charges with the sodium is making the inside of that cell less and less electronegative, which means that cell has the potential to depolarize. Remember, depolarized cells do their job, so we're going to try to depolarize this muscle and have an action potential in that muscle. An action potential in the muscle means the muscle cell is going to do its job, which is to contract. Now, as long as acetylcholine is in that synapse, then it can continue to bind to those ligands or to those receptors. So we have to get rid of that acetylcholine so that this action stops. One way that happens is acetylcholine simply diffuses away. So it moves away from the area where it was secreted and diffuses out into the extracellular environment. The other way that acetylcholine is removed from the synaptic cleft is with an enzyme called acetylcholine esterase. Remember, ACE indicates enzymes. So it breaks down the acetylcholine into acetyl and choline, and then some of the choline is taken back up into that neuron so that it can be repackaged and used again. Alright, so I know that's quite a bit to take in, so let's stop this video here, let you take a little bit to think about what's happening here, because these first steps are very vital to understanding what's going to happen inside this muscle cell in order to get that muscle to contract.